speaker is none other than Dr. Kaushik Sarkar. He is the director of Institute for Malaria and Climate Solutions, IMAX, and Malaria No More initiative. And Kaushik Sarkar, he is also my senior to my uh, alma mater, that is medical college. And he did his MBBS, he did his MBBS from medical college and did his MD community medicine, then executive MBA business analytics, PGP AIML. And he is a global health professional turned artificial intelligence practitioner who aims to benefit billions health in the and Malaria No More India. Besides, he is one of the founder director of SSO Global Health Consultations Limited, India. He has advanced several strategic and technology innovations, including precision programming, climate-based forecasting models to strengthen health systems and access to services by the most marginalized. He also led the establishment of the Strategic Support Unit and convened multi-sectoral platforms, Malaria Action Coalition, and India Interagencies Expert Committee on Malaria and Climate to accelerate the India's progress towards malaria elimination. Our chairperson will be Professor Dr. Rabindranath Rai, Professor, Department of Community Medicine, Medical College, Kolkata.
Hello.
Hariman, your voice is breaking.
Hello.
Hariman, your voice is breaking.
Okay. The connection.
May I audible?
You're audible now.
Hello.
You're audible now.
May I request Professor Dr. Rabindranath Rai sir to give a brief introduction about the session, and please ask the speaker to continue the session. Thank you.
Thank you. Am I audible? Am I audible, please?
Yes, sir.
Yes, sir.
Yes, sir. Okay. So thank you very much for these sessions. Actually, this is a session, as I presume, that it's a innovation. That means what are the new developments that is going to change the malaria situation in the country as well as across the globe. So this is a really extremely important issues that should be dealt in the event of that where we are expecting zero malaria as our goal. So he is a very eminent person to deal with these issues. So we shall be very interested in knowing what are the newer developments and which of the developments can be applied in context of our country particularly. Thank you. I request the speaker to continue. Hello.
Thank you, Chair. Hello. Am I audible, Dr. Roy?
Yes, yes, you are audible, please.
Thank you, sir. Thank you for inviting me to join this particular session. My heartfelt thanks to Dr. Ibu Basu and the organizers of West Bengal IPSM for this wonderful initiative, Dr. Dalui for the intro, and Professor Roy with all the greetings. I'd like to start with the note that malaria elimination is an urgency for the country. And when the Honorable Prime Minister set the pledge in 2015 in the East Asia Summit to eliminate malaria by 2030, we had a 15 years roadmap starting from 2016 to 2030 right after. The first national strategic plan focused on elimination that changed the paradigm from control to elimination came in place in 2017, and that is on the verge of completion now. So the next year, we will see that the national malaria strategic plan will be revamped. Now, essentially, elimination, as we know from the experience of polio, that India needs malaria-free also for 3 years consecutively, and then beyond, to sustain their malaria-free status and receive the malaria elimination certification. So that means the next strategic plan that comes in 2022 and that ends in 2027 should be the endgame strategic plan. Now, when we talk about the innovations per se, every particular innovation, be it a diagnostic, be it a drug, or insecticide, or even strategic innovation, they have their own life cycles of development. Now, there are certain innovations which, and usually we all know that, drug development, it's a 6 to 10 year life cycle, inclusive of the regulatory affairs and the clearances. So I'll talk more about those developments which are newer in the pipeline and which are at the more matured state. And I'll also talk about a little bit of strategic innovations that are taking place across the globe, and we are also focusing on different innovations. So I'll share my screen with the chair's permission. Professor Roy, am I permitted to share my screen, sir? Thank you, sir.
Dr. Tosi, please mute yourself.
Yes, it is clear.
Mute yourself. Nishita, please mute the other person.
Yes, sir. The audio video document.
Dr. Kaushik, please share your screen.
I suppose my screen is visible, right?
Yes, it is visible.
Thank you. So it's playing with some of your settings. It will not focus. Okay, so I'll go by slide because it's showing a little. Let's share. So I believe there's a background sound. Sorry for a background sound that is coming all along continuously. I don't know where from this is coming. I believe if I move by this, the sound will not play. I'm not sure why the sound is coming. So I'll start with the different strategic phases of the innovations. First, let us move to the diagnosis part. So in malaria elimination, there are 3 core strategies, and alongside that, there are cross-cutting strategies. So we start with early diagnosis and complete treatment as the first strategy for malaria elimination. So in the diagnostic phase, the first innovation that came in India after microscopy was the rapid diagnostic tests. The advantage that rapid tests brought to India was the fact that microscopy takes time, and there were delays in blood examinations and getting their results, not for days, but for months. Even now, in certain places of the country, it is much more delayed where reliance on microscopy is more and the inaccessibility is high. So in those areas, rapid test has been recommended, and it is widely used as point of care test because the result comes within 15 to 30 minutes. Now, there are several innovations within the rapid tests as well as point of care tests based on the challenges that we face. The first challenge that we face for any rapid diagnostic test is the lack of sensitivity. So the basic premise is that when we are conducting a particular test, we are conducting in a febrile patient under the routine malaria surveillance. Unfortunately, many of the malaria cases that are there in malaria endemic areas or high endemic areas, they are asymptomatic or afebrile. So that's a population that we are missing. Beyond that, even if we do mass screening with a rapid test or even with blood slides, we miss a number of cases which are detectable by molecular tests like PCR. So that means we are missing then again a whole bunch of cases even after applying mass screening in certain areas. So essentially, the challenge is how to accurately detect the malaria patients. And for that, several new molecular technologies are coming. Even PCRs or TrueNATs, they are being scaled. And now with the COVID-19 pandemic, what we have seen earlier is the massive capacity of scale up of the Indian government of PCR technology or the molecular tests. So first of all, what we need to focus on is the accuracy of the test. The second element is, of course, the sensitivity. The third bit here is the molecular testing ability and the timeliness of those tests. Fourth element is, of course, on the diagnostic bit at the vector side. For that, advanced microscopes are required. Even in the microscopic front, there are several innovations these days. First of all, the initial bit of innovation includes multi-piece microscopes and others. And then, on the other hand, we have training microscopes, which allows, and these days we have microscopes different parasites, but the reliability of such microscopes is still under question. So programmatically accepted innovations include rapid test. Now, ultra-sensitive rapid test, which can detect parasites at a concentration of 1 parasite per microliter of blood, they are also in place, but have not been introduced in the country. We need a fast track mode to introduce those innovations in India, especially in the high endemic areas, and also for the private sector to fast track innovations for bivalent antigen focused elements. For example, if we are talking about the ultrasensitive tests, these are currently for monovalent antigens or Plasmodium falciparum only. Bivalent tests have not been matured, and their life cycle is getting also in terms the development process of the development cycle. The next element I'd come in this piece is treatment bit. So in malaria treatment, the major challenge includes resistance. As we have seen earlier, chloroquine and other drugs, they have been resistant consecutively. Sorry, is there a background?
Nishika, can you please mute others? Nishika, Dr. Sarkar, sorry for the interruption.
No, thank you. So, the next element is the treatment element. In the treatment element, the major challenge is the emerging drug resistance, and we don't have many drugs to spare here. Drugs are under development, as you can see, for new treatments. In the new treatment bit, there are sugar transport inhibitors, Plasmodium falciparum 84 inhibitors, that is the sodium channel inhibitors, the H plus transport inhibitors, lactate as well as B type, DHODH inhibitors like ganaplacide, aquaporin 3 inhibitors, isoprenoid biosynthesis inhibitors are there. And then there are farnesyl transferase and PFP. I'll not go into detail of these drugs, just touch upon how these drugs function, maybe for a couple of instances. So for example, when the Plasmodium parasite enters the RBC, it alters the intracellular environment by changing the permeability of the RBC membrane. And that it does with sodium transporters, with hydrogen transporters, and others. With H plus transporter, it keeps the internal 7.3. Similarly, for sodium transporter, it also changes the sodium permeability. So when it does all of this, what happens is the intracellular environment, although was originally not suitable for the parasite, it becomes quite suitable after it enters the RBC. The inhibitors that they effectively inhibit the transport and levels of innovation of the testings and treatments. So these are at different phases of trials that we see. All of these medicines, Novartis, Takeda, and Medicines for Malaria Venture and several other entities are involved in these trials. Ganaplacide, DF, DHODH inhibitors, phosphatomycin, dihydropypiraquine are some of the new molecules that are emerging. Unfortunately, we still have challenge in terms of formulation, especially for kids. We still have challenge related to radical cure of malaria in pregnant women, infants, and G6PD deficient individuals, mostly tribals, and we still have challenge related to compliance because pharmacovigilance setup or the set ecosystem is has limited capacities. We cannot also introduce the products which may have serious deficiencies. And of course, the emerging drug resistance, especially in the neighboring regions, like the regions of Myanmar, especially, and also in parts of Africa, they are a matter of concern. The next bit is the vector control part, is the insecticidal net. There are different vector control interventions that exist. First of all, it is insecticidal net, or the insecticidal net. That is an adult control measure. Then we have different larval control measure and biolarvicides. So, of course, source reduction is another measure. Another adult control measure is the indoor residual spraying. There are several other pieces. So, vector control is a space where we have seen majority of the innovations happening in around long-lasting insecticidal nets. Previously, it was ITN, insecticide treated, where communities used to gather. They used to dip their own bed nets. Then in a certain concentration of insecticide, and then they used to take that up. That life cycle of that net was 6 months, and the efficacy or the effectiveness of that net was limited. When we are talking about long-lasting, the difference is the polymers or the fibers are impregnated with insecticides. So these insecticides are specifically permethrin or the pyrethroid insecticide. So deltamethrin, alpha cypermethrin, these are the insecticides that are impregnated, and we know that there are emerging resistance across the country, especially for the most prevalent, vector. The Anopheles tulicis, which is causing these challenges in terms of managing the vectors. Now, apart from this, there are newer nets also, like the next gen nets, which we call PBO nets, polyethylene butoxide, which is used to improve the efficacy of the L-lines in different settings. It has not been approved yet in India for by the regulatory authorities for introduction. So in India, long-lasting insecticidal nets have been scaled up in over the past 5 years like anything. And the northeastern state, the state of Odisha, Jharkhand, Chhattisgarh, Madhya Pradesh, and small amounts in Uttar Pradesh and in other parts of the country, they already have these long-lasting insecticidal nets. This is provided only by the government, and it is not provided through the private market. So, and because of several concerns related to the disposal of the nets and, the marketing, the pricing, and all other bits. So these nets are still the mainstay of vector control, and the huge decline that you are seeing is partly contributed because of these long-lasting insecticidal nets as well. Now, there are opportunities in other vector control areas where we are not seeing much innovations. So, sorry. So if we go by indoor residual spraying, larviciding, and source reduction, there are aqua-based or water-based insecticide products that are being innovated. Long-lasting indoor residual sprays or long-lasting insecticides are have been manufactured. Those molecules are also available. They are being tested and tried in different parts of the world. Larviciding, BTI is still there, and there are new approaches of larviciding. New technologies for larviciding is also in force in different parts of the world. For example, in several of the island areas, the drone technology is being used for larviciding, and we have also seen that the drone technology is used widely in the agriculture sector in India for insecticide spraying. So there is an opportunity to introduce drone technologies for larviciding in specific geographies depending on the specific type of vector that exists. So there need to be a proper vector surveillance to support this particular initiative, but at the same time, these technologies are still waiting outside the border of this country, and we need to be inclusive of the overall technological portion of that. The third bit is, of course, the source reduction, which has more to do with the population perception and the community engagement. Certain innovative approaches have been undertaken to improve the source reduction bit. So, for example, under Ayushman Bharat, cleaning drive or village cleaning drive is a common practice. So, just to convert that village cleaning drive or usage of that village cleaning drive for reducing the breeding places can be one of the approaches, and we have found this approach particularly effective in our areas where we don't have to mobilize a lot of people. We just need to mobilize the community under the Ayushman Bharat initiative and the Swachh Bharat Abhiyan, and you just reduce the breeding places altogether. What it requires is a little bit of understanding at the frontline and peripheral care level, care worker level, that where the breeding place exists. So training is definitely important from this perspective, that the frontline health workers, the peripheral workers, irrespective of their educational status, as well as the communities, understand where the breeding place exists. Otherwise, these are simple, easy to adaptable innovations that can take place at any point of time very easily and very smoothly. Now, the next element within this agenda is the, sorry, I think. The next element in this agenda is the data and technology part. So within the data and technology, I'll talk about three elements. First is real-time reporting. Right now, even if we see that India's reporting system is getting changed for malaria and different other diseases, integrated health information platform has been tested and tried, and it is being scaled up, and the trainings are on. But still, real-time reporting is a mechanism where we require mobile devices as well as network connectivity on ground. Even if you provide mobile devices to health workers, real-time reporting is difficult because health workers are not living or residing in areas, and malaria is not happening in areas where real network connectivity is there. So that's a major challenge, and there are several advancements and accelerations that are happening at different front. One of that is you just have temporary storage practices for the data, and once you are in a network area, so if I'm a health worker, I'm going to test certain person, and then that test is getting logged, and I'm coming back to a network sensitive available area, and then the data is getting out. So there also, we in this process, we are actually reducing the delay. Apart from that, when you are ensuring real-time reporting through digital reporting system, you are actually improving the overall quality of surveillance. So that's a side benefit that we have once we introduce real-time reporting or any kind of digital reporting system. AI-based prediction and forecasting tools, this is still, people have apprehensions around this particular technology. AI has been used widely during COVID-19, but still there are suspicions that there may not be precise forecasts that can be provided by artificial intelligence, machine learning, and deep learning both, because of one particular reason: the quality of data. AI is good when you have good amount of quality data. Unfortunately, in real world, quality data is difficult to get. So especially when you have a pen and paper based system, data quality is not consistent or inconsistent. The second challenge is that data, when formats are different, so we are getting data in multiple formats, so then it is difficult to club all those data into a single format in the same frequency and then create the forecast for that. Similarly, risk stratification is another approach where computer vision and or machine vision is being applied. So there are several tools that are being innovated in different parts of the world. There is one tool called ZAP Malaria, and there are similar other tools that exist where healthcare workers are provided with mobile devices and one application which detects to the field, and they captures those images of the breeding sites. They come back, and those are analyzed using computer vision to provide risk stratification of a particular area. Once that risk stratification is done, you can actually apply different types of intervention, inclusive of level, source reduction, level source management, insecticide spraying, as well as case management and surveillance in those settings. So these are the data and technology driven approaches. Without data driven decision making, it is pretty difficult to have any sort of precision programming model introduced in the overall program. The next bit here is the newer innovations as we talk about, and the first one is, as I mentioned, the cross-cutting areas. So in the cross-cutting areas, digital and social media campaigns. So previously we used to talk about, information, education, and communication. So when we talk about information, education, communication, it's like the regular activities of miking or, going about public awareness campaign, or, providing messaging through TV spots or audio spots or radio jingles or something. So these are traditional methods. Now, when we are talking about newer methods, and that needed to come during the time of pandemic, because during the time of pandemic, during the time of lockdown, what happened is you cannot reach to the people. You cannot do any group communication. You cannot congregate those groups initially. So how can you then convey your health messages to a person who is sitting remotely and may or may not have access to different technologies and tools also? What we realized is that mobile devices are gaining their concentration of currency in India, so that the concentration of these devices, the density of these devices in many parts of the country are huge. So digital and social media campaigns, this was again one approach that was people were suspicious about this, apprehensive about this, that whether it will be effective or not. But last year, even we rolled out digital and social media campaign to 150 million only through Facebook, Insta, and other digital and social platforms. Beyond TV spots, we can now go to OTT. So you all see Hotstar, Netflix, and others, and they also exist in rural areas. So we just need to move from our traditional approach of using TV spots to, using something else. Where the network connectivity is not there, messaging can be changed. So messaging can be provided with SMS, messaging can be provided through WhatsApp, messaging can also be provided with other channels, messaging can be provided through teen boards or LED boards. So there are different modalities to provide messaging. We just need to find which particular modality can be useful in a particular area. And the other approach in communication, what we have realized is there is a static messaging that goes. It's a static approach that there is something written or something told that we call a static messaging that goes. And there are opportunities to create dynamic messaging and also create campaign protagonists. So when you deliver a communication through a campaign mode and there is a campaign protagonist that exists who tells the people about the key messages in a storytelling format. So these are certain approaches that needs to be imbibed or integrated to the overall programming. There are targeted intervention approaches to introduce precision programming. So precision programming essentially means that the interventions where they are needed, they need to be applied to those areas. And targeted, that essentially saves a lot of money. So just to give you an example how appropriate targeting saves intervention. So earlier I talked about afebrile malaria or asymptomatic malaria. So afebrile malaria and asymptomatic malaria, they can be detected when you conduct mass screening. Now, let's say, for example, one diagnostic test cost 20 rupees. And you have 100 odd villages where you need to identify malaria. So essentially, by taking an average of 1,000 people, we have 100,000 people to screen for malaria. And the cost is 20 rupees. So that means 20 lakh will be the overall costing for that mass screening intervention. Now, if we know that in high endemic areas where this mass screening need to be applied, so these strategic approaches allow mass screening to be applied maybe just in 20% of these villages. So there we use the same Pareto rule and its advancements that 20% of the population is responsible for 80% of the transmission and they get the most
Koshik, you have been disconnected probably.
It's not audible.
Koshik, you have been probably disconnected.
Dr. Sharkar, are you here?
Sorry, I got disconnected.
Yes, we got it. Please reshare. No issues.
Actually, it got disconnected not because of the mobile, issue. The laptop got crashed, so I won't be able to share the screen, but I'll talk about it. No problem. And sorry for the glitch. So, what happened? In terms of the overall cost effectiveness is when you are actually applying interventions at scale, it is needed to identify the areas where the intervention can be posted. So the cost can be brought down to just 20% of the original cost or maybe 30% of the original cost if targeted interventions can be applied in different settings. What I cannot show you at this point of time, but I'll talk about it, is the next element, that is vaccine. Everyone is talking about it, and there are different types of vaccines that are being tested and tried, and there are other preventive methods as well. So the first element in vaccine is we have just seen, WHO announcing RTS-AA01 vaccine, and it's not a new technology per se. Vaccine testing started in malaria in 1945. The first malaria vaccine was against the P. leufori. This was having the adjuvant of formalin as well as killed cock bacilli, which by the name of the researchers, Fraud's adjuvant, the adjuvant was used to be called. So the major challenge was for this particular vaccine was the reaction of the adjuvant. Then this was tried, for rhesus monkey, for the zoonotic malaria, Plasmodium knowlesi, and the same adjuvant reaction persisted. So the major challenge was how to overcome that. And decades later, alum, an adjuvant which was first tested in 1935 or 1930s, that became the adjuvant for malaria vaccine. In the initial decades, malaria vaccine, RTS, the circumsporozoite protein and its targets, and the different accelerations that have happened. So it took decades to find out, and still we have a vaccine that is 30 to 50% effective, and maybe a little more effective in specific target groups. But it has limited effectiveness, and it can only be applied in high endemic areas. So that is the major drawback with the current vaccine. There are many other vaccines in the pipeline as well. For example, R21, it's in phase 2. There are, so just to summarize that there are 3 different types of vaccines that exist, malaria vaccines that exist, mostly in different stage 1 or phase 1 and phase 2 stages of trial. One is the pre-erythrocytic, then the transmission blocking and sexual and asexual stage affecting types of vaccines. So these vaccines, depending on which particular category they target different parts of the parasite and different different receptors and different proteins, PFS and circumsporozoite proteins, and there are several other proteins that are targeted by the vaccines. The next gen vaccine are focusing on multi-stages. So what has been seen is that the current generation of vaccine, they have limited effectiveness because they target only single stage. The next gen vaccine, because they have multiple stages, they can target multiple stages, the effectiveness can be more. So they kind of use the same parlance of probability theory, where reducing the probability by geometric progression actually reduces the overall chances of infection. Another technology that is not being talked about currently, but it is still in place, and early phase trials have been there with promising results, are the monoclonal antibodies. So monoclonal antibodies have been successfully used in early phase trials, where it has shown to remove or reduce, obliterate or parasitemia. So these antibodies can actually reduce parasitemia, prevent infection. So even after introducing infection, they are not allowing the parasite to come into the blood and actually clearing the parasite. So these monoclonal antibodies are promising technology for prevention of malaria. In the vector side also, there are newer molecular advancements that are happening. Genetically modified mosquitoes and others are being tested and tried. But one concern is definitely there regarding the ethical clearance, the ethical concern. So there are thousands of mosquito species that exist, but only a few, less than 10 species are actually important from the perspective of malaria elimination. So protecting the environment and the food chain and the ecosystem is important. So we cannot bring a technology which can harm the overall ecosystem. So that's why these technologies, even if you go by WHO's site, they are saying that these technologies help in providing solutions, but they are still under review of ethical clearance and for concerns. All of these I have talked about different technologies, tools regarding the innovations. innovations exist within the strategies also, which we don't focus. I mean, programmatically, you see the same strategies exist year after year. People are talking about newer technologies and tool, but I would say that hundreds of years have passed. Smaller strategic innovations can do a lot more impact, or can produce a lot more impact compared to, waiting for newer technologies, innovations, and just trying for when these technologies would come. So these are the, and there are several examples, but in the interest of time, I believe there are only 10 minutes left. So I'll pause here and request to, the chair to have his address, and any particular question, I'm happy to answer.
Thank you, Dr. Sharkar. It's an elaborate and extensive research work, I guess, that you have made, and this is an interesting for all of us. So I do request our chairperson, Professor Dr. Rabindranath Rai, to comment or to invite any questions from his side.
Thank you very much. This is a wonderful sessions, really. We have been enlightened with so many new developments which can change the, but one concluding remarks, the speaker said that the same strategies or the conventional things we are going for a long time. In fact, it's not changing. So it would have been very highlighted some of the examples so that what are the any technology which we use for a particular area or a country, it should be really need-based as well as it should be affordable one, that we have to keep in our mind. So keeping that in mind, there are which technologies or which newer development we can use in our context of India. That will be very particularly, we have to review that. If speakers can highlight on any audience can have questions regarding that. The speaker has said almost in all areas, like the diagnosis, treatment, vector control issues, then behavioral change issues. Then again, but one thing is very important issue, the environmental modification. Whether the environmental modifications, any newer things are coming, whether this can be done. Particularly this is in the urban area, we know a lot of environmental issues is really related to malaria. So I shall ask if anybody have a questions on that issues, he can ask the questions to the audience, to the speaker. Okay. So
Dr. Sharkar, sir, already Dr.
Yes,
Dr. Patnaik has asked some question regarding insecticide treated bed nets. Is it available in the market? A few days back, there is a case in Kolkata Municipal Corporation. There is a reaction due to using of the mosquito, the insecticide treated bed nets, and from then the severe skin rashes and everything came out, and there is a question of using it in a mass scale in the slum area. So what's about your comment, and as well as what's your concern regarding this?
Yeah, so as I mentioned earlier, insecticide treated net is a different thing. I mean, it's the older technology where you just dip one a common bed net into insecticide, take it up, and the insecticide get layers on top of the bed net. And yes, there are adverse reactions like this, not widespread, because this technology has been used earlier also. If someone has allergic reactions to it, they can have these challenges. There are other, smaller adverse or minor adverse reactions as well. For example, burning sensation in eye, itching, and others. So these sensations also persist. But ITN has been used in the past. I mean, in the past decade only, 2010, and even before that, ITNs were used. Long-lasting insecticidal nets, they are being used for a purpose, and where the life cycle differs. It's not for 6 months. Long-lasting insecticidal net persists for 3 years or 20 washes. So that lifespan is really important when we give some commodity to the community. These are not available in private market. ITN, the bed nets are available in private market. People are purchasing it. Insecticides are available, deeping it and creating the ITNs. But LLINs are company manufactured impregnated nets. So they only come with insecticide impregnations and they are not available in private market. Government of India provides states based on their demands and the programmatic norm. Only subcenters more than one API are eligible to receive long-lasting insecticidal nets. And these nets are given in rural areas and distributed both through domestic budget as well as global fund support by the government to the state governments, and then the states provide those nets to the or distribute those nets to the communities. Till date, northeastern states, the state of Jharkhand, Chhattisgarh, Odisha, Madhya Pradesh, some amount in UP, Gujarat. So these areas have received long-lasting insecticidal nets, and I believe the government is scaling up this particular intervention. And because of environmental concern, and there are a few other concerns as well, it is not coming into the private market. So government has taken the responsibility of supplying it across the country and scaling it.
Thank you. Thank you so much. So this is a genuine question from the buyer side also. So I would like to ask another question on behalf of Dr. Sinjita Dotto. She has mentioned that what changes or innovations in malaria eradication strategy is required at present. So your brief.
So first of all, if you go by the National Framework for Malaria Elimination, it says that district as an unit of implementation. And the last strategic plan actually stratified the district for malaria elimination and interventions accordingly. Now, with reduced transmission, several changes are required. For example, the indicators of malaria program and they need to change. Simply because previously we used to have the 3 years average to detect outbreaks and others. The cases have gone down. So malaria metric needs to change. The second element that needs to be changed is the assessment of burden and the surveillance approaches. As we move towards elimination, there need to be contact tracing, there need to be detection of migrant as well as newer local cases, and also local transmission in the pregnant women and children. So the surveillance approaches need a lot of change. Vector control strategies, majority of the vector control interventions are only applied for areas where API is more than 1 or more than 2, especially the adult vector control interventions. So in that sense, there need to be strategies to scale up larval reductions, and at the same time, there are opportunities to create insecticide resistance management strategies which is not there, so that the growing emergence of triple resistance to insecticides, they can be reduced. I just talked about 3 of the maybe 100 challenges that exist. We need new drugs for Plasmodium vivax, that is not there. We need new strategies for pharmacovigilance, that is not there. So the list is huge. We conducted a complete series on that, and happy to provide more information over email or so, because just to summarize, each and every strategy for end game will possibly be completely different than what we have right now.
Thank you. So I'm just going to ask you the last question from our participants. So that is a brief ideation about when the malaria vaccine can come. You have elaborated extensively. Still, just 1 or 2 liner on that. So when do you expect about this malaria vaccine in India?
We don't even expect malaria vaccine in India right now, because to have that expectation, the technical expert committee of the Indian government needs to sit together. And right now, WHO has only approved. The Indian regulatory body needs to approve it for use in India. And post that, the areas where this vaccine can be effective, because this vaccine is not widely effective, where it can be effective, they need to be identified. So it requires time.
Okay, thank you so much. I think we have all covered the malaria and recent innovations, and you have covered it very well. And thank you to our chairperson for sharing this session out and gave his important comment on that. Sir,
I have one question, please. May I ask a questions again for a clarification? Can
Sure.
I? So one thing you said the contact tracing.
yes, sir. Yes,
So,
sir,
you
please.
know, this is being India is being a endemic country, and there are a lot of cases, you know, and this is being a vector-borne transmission mainly. So how this contact tracing is really be useful here?
Yes, I was expecting this particular question. In malaria, they call it case-based surveillance. So it's very similar to contact tracing. So it's not a human-to-human contact. Rather, when an infection is traveling from a particular geography. So for example, I'll take an example, inter-district example in the southern part of Odisha. So the southern border of Odisha and the Andhra Pradesh border or the Chhattisgarh border, they are adjacent. Now, as the cases are reducing in Odisha, while the cases are still persisting in those borders of Chhattisgarh and Andhra Pradesh, these cases are, and there are migrant workers, they are traveling. When they are coming in or going back to those particular geographies, they need to be tracked wherever they are staying for the last 13 or 15 days. So that is called contact tracing or case-based surveillance, and there are other names also. I mean, T4 strategy, T3 strategy, all are based on these parlances. What is needed is to identify that where this person has actually stayed and infected the vectors and created a local foci when this person has moved from one geography to another. So if a geography has been cleared of malaria parasite, both the vector as well as the human, and a new case is coming to a particular site, it is important that the mosquito population does not again get the parasite pool inside them, and the human population does not again get the parasite pool through those infected mosquitoes. So in that sense, contact tracing and tracking of the cases, they are really important.
So thank you very much
Sir.
for this wonderful session again. So I think
Yes,
it
sir.
can
Thank you.
be now.
Thank you, sir. Thank you. My
Okay.
highest
Thank you.
regards. And thank you, Dr. Ibu and IPSM for this opportunity.
Thank you, both the speakers.